Hi all, I'm Jorge. I'm a research fellow here in Emmanuel College, and I'm going to tell you about my research, which is focused on genome organization. So let me just give you an example of how complex and challenging can be genome organization by saying that our DNA is about 2 meters long, and it needs to be accommodated in a very tiny space, which is the nucleus of the cell, which has a size of about a micron. So that task is not just difficult because of the size of trying to accommodate some polymer that is so long as DNA, about 2 meters in a so tiny space, but also that is crucial how the DNA is folded, because depending on how the DNA is folded, we lead the cells to work completely different, and then the cells will work as neurons or as organ cells or tissue cells. So as you can see, it's crucial to understand properly this folding and genome organization of our DNA. But also because fully understanding of that will allow us to know more about DNA-related diseases as Alzheimer's or cancer, and to improve our knowledge about cell diversity, the growth of cells in embryos, or what happens when we are getting older and our cells starts to malfunction because of the aging. So the tool that I'm using to investigate genome organization are molecular simulations. And I chose molecular simulations because it allows us to investigate the different thermodynamic and physical determinants that are behind this behavior, and also because they provide us a molecular insight and also knowledge about the key molecular interactions governing these processes. So I'm going to show you an example now of how these molecular simulations of biological processes like genome organization look like. Here we have an example of a molecular simulation of a DNA high-resolution model in presence with several histones, and what we can learn are the molecular interactions governing this association, as well as the mechanical and thermodynamic driving forces present in here. It has been proposed recently a new paradigm in genome organization, which has been called liquid-liquid phase separation. So this is exactly what happens when we have in a glass oil and water, that even though both of them are liquids, they don't mix each other. So this is what also has been proposed happening in cells in order to determine the different organization of the different compartments. So what we believe until now is that these different compartments, they were separated by means of physical membranes, but this paradigm, what it proposes is that there is no need of membranes for organizing the different compartments, and even though they are liquid-like, they can coexist in chemical equilibrium by means of this liquid phase separation process. So in my research, I'm mostly focused on characterizing by means of computer simulation this process for pure proteins and also for proteins in presence of RNA and DNA. Here in this simulation, we have the human protein phase in presence of a single strand of RNA, this dark blue chain, and as you can see from an homogeneous phase, where everything is spread, the RNA is able to organize all the proteins into a single condensed phase. It has been found that some amyloid formation diseases, as Alzheimer's, are due to the jellification of the proteins forming these liquid-like compartments, because they cannot behave as a liquid anymore while they age. So in my research, we are developing new models to understand this behavior. Here in this simulation, we have a new model that we have designed in order to capture this aging process leading to the amyloid formation. By partially modifying the sequence of these proteins, we avoid this aging process and we enable the proteins to diffuse enough to form these spherical liquid-like compartments. So as you can see, with computer simulations, we are able to design and to develop models in which we can specifically modify and mutate parts of the proteins in order to avoid the formation of these amyloid regions, which are related to very dangerous diseases, or also we can modify part of the proteins to enhance the formation of these protein condensates and these liquid-like organelles where essential chemical reactions for the cells happens there. So I hope you have enjoyed this brief update of my research, and I hope also that you are all doing very well. So bye-bye.